last decade, my lab has been working on 4 lines of research which are interconnected. The first one is Biomolecular Corona, where we basically try to identify and introduce overlooked factors at the nanobiointerfaces. And the ultimate goal here is to enhance the safety and therapeutic efficacy of nanotechnologies, and also to facilitate their successful clinical translation. The second part is regenerative medicine, where we basically develop engineered nanotechnologies for heart regeneration and wound healing applications. The third one is stem cell nanoengineering, where we basically try to develop smart substrates that can mimic the shape and surface properties of mature cells to basically help them to physically direct their differentiation toward any type of mature cells. And the last topic is the optoelectronic nodes, which we basically try to mimic human olfactory system for detection of disease at their very earliest stages. So among this research, I'm going to talk about the potential and challenges of biomolecular corona in nanomedicine. So as we all may agree on, nanotechnologies are now being used in many products related to non-biomedical applications. By contrast, and unfortunately, the use of therapeutic nanoparticles in medicine has been quite limited. And many of the potential products, specifically therapeutic nanoparticles by leading companies, got failed in clinical trials. So as a result, there are fewer than expected FDA-approved products out there in market, like Abraxane, which is a protein conjugate with paclitaxel. It's initially being used for treatment of breast cancer, but now it's being used for like other type of cancers as well. The other example is ferumoxytil, which is a dextran-coated iron oxide nanoparticles, and it's being used for iron deficiency applications. So the reason of having this big gap in the marketing capacity of nanoparticles for medical and non-medical applications is that basically the very same qualities that make nanoparticles so distinguished for non-medical applications also makes them toxic and unpredictable when being considered in medical context. So in order to minimize this gap, there are many questions in the field which needs to be properly addressed. So among the pool of those questions, we have been working in addressing 3 main questions, which are like why there is significant conflicts in the biological data and even exact same type of nanoparticles. The second one is that why there is a huge gap between the in vitro and in vivo results in terms of the safety and therapeutic efficacy of nanoparticles. And the last one is that why the field suffers from a huge misinterpretation of the biological outcome of nanotechnologies, which basically causes misprediction of their biological fates. So we believe that there's one main answer to this question, and it's that there are several hidden factors at the nanobio interfaces which had been widely ignored during last decade. So we believe that there's an urgent need for systematic debugging of the current protocols and approaches in nanobio interfaces. So this scheme basically shows the list of identified and reported overlooked factors in the field, which are divided in 3 main categories. As you see, the nanoparameters, the nanobioparameters, and the biosystem elements. And these factors should be carefully considered and reported in nanomedicine protocols to basically create a robust and error-free outcomes. A clear example is that, like you know, the conventional toxicity assays should be modified for nanotoxicology evaluation. And the reason is that basically most of the toxicological assays that were created were basically that are being used in the current like nanotoxicology field have been developed for like toxicity evaluation of chemical solubles and not for nanoparticles. So therefore, if we need to use these approaches or these assays for nanotoxicological purposes, we should apply essential modifications on the desired assays. Otherwise, we may like add more unintentional errors to the current literature. So as the big chunk of the nanobio parameters is occupied by protein corona, I'm going to talk about the role of biomolecular corona. So the biomolecular corona or protein corona happens when nanoscale objects get in contact with the biological fluids. And its surfaces basically gets covered by a layer composed of a variety of biomolecules. Most of them are proteins, but we also have like lipids, metabolomes, and other type of biomolecules, which we call this layer a biomolecular corona. So the protein corona basically gives nanoparticles a new identity, which is called biological identity, which is totally different from this like synthetic identity. So in principle, we have two different types of corona. It's a soft corona and hard coronas. The soft corona is formed within a few seconds after the interaction of nanoparticles with protein environment. These are reversibly and rather loosely attached proteins, but this layer would be evolved to like hard corona, which is like more irreversible and tightly attached proteins. What the cells actually see is the hard corona coated nanoparticles, and the composition, type, amount, and decoration of the protein corona can basically dictate their interactions with cell receptor and defines the biological identity of nanoparticles. So it's very hard to see 3D structure of the protein corona. We have like a couple of techniques like DLS, electron microscopy, differential centrifugal sedimentation to basically indirectly measure the protein corona. But if you want to see them directly, there are few approaches. Like one of them is cryo-TEM. And as you see here, we see the 3D constructed images of polystyrene nanoparticles without corona, and after interactions with proteins, this would be like the corona-coated nanoparticles, which we can get 3D information about them. So with the help of some image analysis, we can basically see the 3D structure of like each of the nanoparticles and see the corona shape. So like I mentioned earlier, the biomolecular corona composition can dictate the biological fate of nanoparticles. So for example, if a complement proteins and opsonin-based proteins are participated in the corona composition, the particles would be removed by immune system and will end up in like liver and spleen. If carrier proteins like albumin have rich contribution in the corona composition, then we can expect to have like high blood circulation time, which is good for like the use of targeted nanoparticles. If like other specific proteins like ApoE come into the composition of the protein corona, then the particles have the capacity to pass a specific barriers like blood-brain barrier and like target the brain tissue. Overall, the biomolecular corona field induces several issues on the central goals of nanomedicine. This scheme shows like the major issues presented by the biomolecular corona layer. For example, the corona can shield the targeting species at the surface of nanoparticles and therefore can cause mistargeting. The corona can also add another layer of barrier to nanocarriers and alter the drug release profile. And like the other problem is that the formation of biomolecular corona can change the nutrition balance of the cell culture media and induce like false data to the toxicological outcomes. So let's talk about how protein corona enhances mistargeting of nanoparticles. So as you know, one strategy to obtain a high targeting yield in nanoparticles is to functionalize their surface with targeting species that basically help nanoparticles to bind to the cell receptors of the desired cells and facilitate like nanoparticles uptake through several mechanisms like receptor mediated endocytosis. So in vitro results generally confirm the high capacity of those targeted nanoparticles to the desired cells. But when we come to the in vivo condition, we see much lower targeting efficacy and unfavorable distribution of nanoparticles, which is basically one of the main reasons for limited success of targeted nanoparticles in clinic. So we and others proposed that the discrepancy between these in vitro and in vivo targeting results is at least in large part related to the shielding effect of biomolecular corona. So to test this idea, we developed targeted silica nanoparticles, the BCN functionalized silica nanoparticle, and probed their interactions with azide-coated silicon wafer substrates. So the copper-free click reaction here between the functionalized nanoparticles and the azide groups at the surface of silica substrates was used as a potential model targeting reaction. So we used both corona-free and corona-coated nanoparticles to evaluate the effect of biomolecular corona on targeting efficacy of these nanoparticles to the functionalized substrates using fluorescence microscopy. And we found that the formation of biomolecular corona can shield the targeting ligand at the surface of particles and can significantly reduce the targeting efficacy, like by 90%. So other groups showed the same effects on the cellular targeting of nanoparticles. So to overcome this critical issue, we proposed two approaches. The first approach is to pre-coat nanoparticles with specific proteins, which has a capacity to enhance enrichment of targeting ligands with during the biomolecular corona formation. So to test this first strategy, we used gamma globulin as a pre-coated protein to absorb more of its type proteins due to protein-protein interaction. Like we predicted that we can get more immunoglobulin and activated complement factors to the composition of the protein corona. And for the targeting purposes, we selected macrophages as a model cells, mainly because the participation of the immunoglobulins and complement proteins in the protein corona can accelerate their interactions with macrophages because they have Fc receptors. So we did liquid chromatography mass spectroscopy, and we compared the protein corona on the surface of bare nanoparticles and nanoparticles with pre-coated gamma globulins. And as you see from the results, the pre-coated nanoparticles demonstrated substantial contribution of complement factors and immunoglobulins in their corona structure compared to the bare nanoparticles. We also followed their cellular uptake and found that basically the pre-coating process had a great capacity to increase the uptake of nanoparticles. And as you see in the confocal images, the pre-coated nanoparticles show higher co-localization of Fc receptors compared to the pristine nanoparticles, confirming that the pre-coating process can enrich the targeting efficacy of nanoparticles through recruitment of the proteins with natural targeting capacity. So the second strategy is to get rid of the shielding effect of protein corona by using specific coatings that can reduce the corona formation. So conventionally people used PEG, but then they found that PEG can like increase binding of nanoparticles to the specific type of protein, so the shielding cannot be affected much. But one good candidate is Zwitter ionic compounds, which basically limit the formation of the protein corona. So in this regard, we have designed the biotin cysteine conjugated silicon nanoparticles, and we employed like biotin as a targeting molecule and cysteine as a Zwitter ionic ligands. So we did the gel electrophoresis analysis to consider the formation of protein corona, and as expected, we found that the formation of cysteine in the coating could inhibit the formation of protein corona at the surface of nanoparticles. And then we performed cellular studies on various cells with or without receptors for biotin, and we found that basically existence of the cysteine can substantially increase the targeting capacity of the nanoparticles in a complex media. There is another approach which is introduced by Dr. Lanfester's group in Max Planck Institute, revealing that changing the mode of interaction of targeting a species with the surface of nanoparticles is also critical for the targeting purposes. If by using like antibodies interaction with nanoparticles through pre-absorption, we can see like much better targeting compared to the fact that we like use chemical conjugation to attach antibody to the surface of nanoparticles. So pre-absorption would be like a better idea. So our current efforts in the field of biomolecular corona are focused on the concept of disease-specific protein corona, which we introduced the concept in 2014. As you know, like the composition of biomolecules in human plasma is strongly dependent on the type of disease or condition of the person, like its age, its sex, or like by condition, I mean like existence of pregnancy and so forth. So based on this knowledge, we speculated that the biomolecular corona of these plasma proteins could be also different in various diseases and various conditions. So in order to initially test the hypothesis, we employed two commercially available safe nanoparticles, polystyrene and silicon nanoparticles, and incubated them with a wide range of human plasmas of various disease and conditions. And then we ran the STS page, and we conducted the mass spectroscopy, and we basically found significant differences between protein composition in the corona shell of these nanoparticles. It's very interesting because for the exact same type of nanoparticles and the exact same composition, the nanoparticles have different biological identity. And this is very interesting because then one can expect that using exact same type of nanoparticles can end up with different safety and biological efficacy in different people, different patients. So we need to develop disease-specific nanoparticles, if not the personalized nanoparticles, to make sure that like the we can have a safe and successful, efficient nanoparticles for disease treatment. So this would be a big challenge for therapeutic applications of nanoparticles, but at the same time, it can provide a unique opportunity for disease detection, because using the same nanoparticles with different coronas, you can look for specific biomarkers or a specific protein patterns that is related to a specific type of disease. So in order to get more robust data, we combine the concepts of protein corona and sensor array technology. And for sensor array, as you may aware of, basically the idea is that you don't develop a sensor based on a specific interaction of your target and the chemical dyes that you use. The detection sensitivity, specificity, and prediction capacity comes from a pattern recognition. So in order to do the same thing for disease detection, we used 3 different types of liposomes, and then we incubated them with plasma of healthy individuals and cancer patients. We selected like 5 different types of cancer plasmas, and then we probed the protein corona formation and analyzed the type of the corona in each cases through like over 1,000 LC mass analysis, and analyzed them with classifiers, supervised classifiers, to see whether we can use this technology for cancer detection. So this is the model that we used. We basically used a supervised PLSDA model based on the protein corona data. And we also used a couple of like other supervised classifiers like CPAN, and unsupervised one like hierarchical cluster analysis, to see whether we can identify the cancers based on the protein corona profiles that we get in combination with the like sensory nanoparticles. And here is the outcome, which is very promising. We could identify different types of cancers through different like classifications. And in order to make sure that we got a reasonable data set, we also used the cohort plasma samples. And by cohort plasma samples, I mean the samples from patients that were healthy at the time of plasma collection, but then developed different type of diseases over the time. So these plasmas were used and were run in the system, in the protein corona sensor array system, and we could identify those like types of cancers ahead of time in terms of like detection time, almost two years before any type of like advanced technologies can detect those kinds of cancers. So I'd like to summarize the points that I've made in this talk. So the consideration of the nanobiointerfaces, and in particular biomolecular corona, is an essential step for getting nanoparticles in clinical stage. And I would also like to remind that the protein corona is one part of the nanobio interactions. We need to consider other factors as well. We also need to consider the biological systems carefully. We need to like report the passage of the cells that we use, the sex of the cells that we use, the origin of the cells that we get, and all those kinds of important like data and parameters should be reported in the nanomedicine related publications. The other thing is that the personalized protein corona should be considered in the nanoparticles design and development. Like I mentioned, the exact same nanoparticles with the exact same synthetic biology can end up with different biological identity. And this means that the same nanoparticles may have different safety and different therapeutic efficacy in different patients. And the last part is that the protein corona and biomolecular corona can induce issues for therapeutic nanoparticles, but at the same time, it can provide a unique opportunity for sensor development and disease detection applications.